some of the live challenges to do with drug discovery, in particular, making sense of the huge amount of data that's out there using AI techniques. So could you switch to the slides, please? There we are. Thank you very much. So just go there. There we are. So and is it possible to get rid of the band at the bottom? There we are. Perfect. So I work for the MDC, the Medicines Discovery Catapult. We're a national facility connecting and collaborating with the UK life science drug discovery community. You've just seen an example of how we're working with a local Manchester company, Chief AI, in the area of some AI services and models. We're funded by Innovate UK and UKRI, and above that, I guess, BES. And our primary focus is to support small companies and translational academics. This sort of valley of death between academic science and the market for new medicines. So the first area, just really skip through very quickly, are a series of tools we're building around collaborative intelligence. And it's actually quite difficult to find the best collaborators if you have a particular wish to find someone with some skills. I guess we're all familiar with dating apps like Tinder and so forth, and often you don't see the full person. You just see a sort of carefully faceted view of that person. We've built some tools that are very objective in finding evidence behind capabilities and skills for particular organizations. We originally planned to do this through a little bit of manual curation, so building a list of companies and so forth, but it's really expensive and you just can't keep on top of that and keep it up to date. So we built a system called CRISPy, which is a live knowledge graph built from the internet of UK drug discovery assets and capability. And at the moment, this is an internal MDC tool, but if people are interested, you can certainly get in touch and we can provide some demos or whatever. So we do very automated gathering of data in the same way that a whole bunch of government agencies do with the sort of heartbeat and status of the UK economy. So we bring in data from Companies House, Charities Commission, from patents, full text patents, published scientific papers, grant applications, and so forth. Load all of this data into a big document lake, and then use named entity recognition, natural language processing, and then word2vec to provide some semantic search capabilities on top of it. This is a view of the interface. You can see very Google-like. We call the tool CRISPy. Type in a term. This is a particular scientific technique. On the basis of the word2vec, semantic similarity sort of tools, using a model we've trained on a large corpus of scientific literature. We can find, of course, synonyms of surface plasmon resonance, SPR, for example, but also automagically find the related concepts and tools that people can then search. And then you end up with a search page, which looks a lot like Google. We did a little bit of UX and people said, I really want something that's very familiar to me, and this is a familiar type interface. But it's a very focused, semantically aware, AI aware search engine across the UK life science sector, indexing up a whole set of documents that typically aren't found in search engines. The main area, though, that I wanted to talk about was this concept of sort of assays in drug discovery. So how do we test drugs? And we heard Waqar talk about assays in the previous presentation. So these are the sort of acid test. Does a drug work in a particular system? And it's interesting to sort of map the historical development of drugs to historical timeline and see the impact of technology on this, but also the impact of technology on the success and cost of drug discovery as well. So in the olden days, the really olden days, people used to do clinical trials. Not that all the subjects were consented at that time, but we used to try various plant extracts and so forth on people. And sometimes that was great. We discovered drugs like aspirin and quinine, and we've heard a lovely sort of story around the sort of unlocked knowledge around artemisinin for malaria. But it's very expensive, unethical to test just arbitrarily synthesized compounds and so forth. And so over time, people started to realize that there were similarities between animal models and animal physiology and human diseases. So we started to use animal models. These were faster, safer, and cheaper, but less predictive. So not every compound that cured a mouse of cancer went on and cured a human of cancer, because there's clearly differences between the model system and the human clinical trial. But a human clinical trial is an assay, an experimental test in an animal model is an assay as well. As we started to understand biology and medicine at a deeper level, we could start to isolate organs and do tests on isolated organs or ex vivo, things derived from a body. These are higher throughput and cheaper. They give mechanistic insights, but are less predictive. And you can see this trend continuing. We've probably all heard about the impact of cell lines and cell line technology on drug discovery. So we worked out, or scientists worked out, that they could grow cells, successful mammalian cells, human cells sometimes, outside of a living organism. And you could run things cheaper and higher throughput, but they were, again, inexorably less predictive. And then finally, in the 1970s and 80s, along came the genome and recombinant DNA technology and high throughput screening. So you could run thousands, hundreds of thousands, and now millions of assays on a particular target, part of the body you're trying to interfere with, but less predictive. So to give you an idea of the costs, here you're probably talking about 0.1 euro, 10 pence per assay data point at this end, and probably closer to 100 million per assay data point here for one compound in a human clinical trial. So an exponential growth in cost as you move across this network or cascade of assays. But drug discovery now is primarily done here in the protein space, and then the compounds are tested in a series of assays going from cell-based to functional to in vivo and then to a human clinical trial. Of course, a drug needs to be safe as well, and there's a series of parallel assays for this. So we started to model all the world's assays, organizing all of these experiments that people have done and you could do, and we extracted these using text mining from the literature, or we imputed them from experiments that could be done on the genome. And so there's a sort of a connection or a path between every single gene or protein in the human body and every possible disease. And one way to view drug discovery is unwrapping or finding the connection between these assays as you go from simple high throughput through to expensive and predictive. So we've spent quite a lot of time building this network and then building some analytics tools on top of this, and we call it AssayNet. Here's a real example: a drug, an anti-inflammatory drug. These are the sort of typical assays that it's tested in before it's used in, say, a gout patient to treat pain. But you can't go directly from here to the clinical trial because it's too risky and failure is too common. This is one path. We extract these paths from the literature and from integration of resources like clinical trials and drug databases and so forth. Drug repositioning is often finding different clinical uses for a drug, different diseases that the same drug can be used in. And often you find an intermediate assay that connects both this test here and a number of different diseases. So this approach really starts to get to the bottom of defining or building a map of mechanism and disease biology from unstructured data. Often multiple targets can be used, multiple parts of the genome targets can be used to treat the same disease. So this is a hypothetical case here where lots of different targets are now known to be able to treat type 2 diabetes, and they show some level of convergence or common activity as you move from the genome through to clinical trials. And one way of thinking about this problem for those of you interested in algorithms is it's really a sort of inverse ant colony optimization problem. So we've got this sort of search space, and what we're using is the literature and the connections between these nodes or these assays that we uncover from co-occurrence in papers to build the solution to this ant colony optimization, where ants will miraculously find the best path or the optimal path between food and their nest. So there's a lot of different computer science and AI techniques used in building this this system that we call AssayNet. One of the primary ones is a database that I built some time ago, now public domain, which extracts from the literature relationships between compounds and targets. So you can search with a target, you can search with a compound, and because of a whole bunch of very well-developed and reliable and robust search tools or similarity tools for both small molecule similarity screening and for target screening, you can generalize this relationship and propose new compounds active against new targets and so forth. But for the purpose here, what we're primarily doing is linking two different assays, a target-based assay and in this case an in vivo assay. This is an example of some clustering where what we're doing is we're taking drugs, we're looking at co-occurrence of assays that connect them. So these are drugs tested in the same classes of assays, and you can see we're starting to provide some clustering or some similarity of the subnodes on this very complicated network. This is again a more complicated hairball type diagram to show that as you scale the network, you still find non-random or very directed paths through this complicated sort of pseudo neural network, the nodes connecting targets through to clinical trials. And then you can go and annotate these points with particular features like family membership and so forth. It turns out that the genes in your body are sort of often relatives of each other through evolution. Different proteins evolve around a common framework, and these often have shared similar function. This is an example here where we've scaled across the various levels of complexity from target-based assays, and then we see convergence around particular mechanistic features through to common activity in an animal disease model, in this case for type 2 diabetes. The other interesting thing you can start to do with this data is learning some of the decision thresholds that people take. So how good does a compound need to be active in a biochemical assay, this thing with a little white squirrely thing on the left-hand side, before someone thinks it's good enough to go into a cell-based assay, this little blob diagram to the right of the paler blue circle. And we can build some activity cutoffs or performance criteria automagically from text mining or from running analytics approaches across the network. Turns out what we were hoping to find was some magic numbers like 7.42 and so forth, but often humans like powers of 10 in making these decision thresholds on real world data, or at least in the data that we have to hand so far. Our longer term view is to start to use techniques such as Bayesian networks across this data in order to learn more rationally these decisions, in particular the combination of properties that are required for a good drug. The last section is really to do with some text mining we've done. So this is a typical description of a compound activity. This is an anti-inflammatory assay in a rat model. There's some jargon in there. So SC means subcutaneous. So we've built a series of specialist dictionaries for this. We've gone through and semantically tagged these particular objects with a sort of ontology. We've done parts of speech analysis in order to understand the relationships between the various terms and concepts. And then we've built a map, a principal component map of the similarity of all, where every single in vivo assay, so animal-based assay, used in the development of drugs. And this is the map. The darker the green, the more data points we have in this particular cell. And this is, of course, an unsupervised method. So what does it mean? What it means is that when we validate this by painting labels of known, say, anti-epileptic drugs, anti-inflammatories, anti-neoplastics, so anti-cancers, diabetes, hypertensives, and so forth onto this projection, this PCA, we see quite discrete clustering. And so we've now got an engine, a text mining engine that can read text that contains bioassay descriptions and classify the likely disease use of those in a very general way. Some areas of biology, so analgesia is a very complex sort of physiological outcome. So this is the suppression of pain. It doesn't project very well into this latent assay space, and probably there's a series of, what this means maybe is that actually there's a series of better words or subclasses or clusters of pain mechanisms that actually need developing. So AssayNet is our sort of ImageNet equivalent, but based around bioassays, one of the fundamental objects used in drug discovery, the tests of drugs. And hopefully I've shown you in sort of outline mode how we go from raw text, structure the data, we add value, a little bit of curation, and we learn some general rules that are useful for drug discovery. This work comes from a number of professional positions I've held. So the ChEMBL group at EMBL-EBI based on the Sanger campus in Hingston for a lot of the database work. Mark Davies and Magda in particular for some of the AI and network graph topology stuff, and then my colleagues at the Medicines Discovery Catapult for some of the more recent work in this area. So thank you very much.